Folks, today we will discuss electrophoresis and advanced biochemical techniques. Um, so, um, in this lecture, we will see that the introduction of electrophoresis, then theory and basic concepts that uh, electrophoresis is used to separate charged molecules based upon their, for example, molecular weight, uh, and the equation that is governing the electrophoresis, that is V is equal to EQ over F, in which V is velocity of the molecule, while E is electrical field that is applied, while Q is the charge on the molecule, while F is frictional coefficient. Then we will see that what are the basic concepts. For example, when we load the charged molecules like DNA and proteins uh, in a buffer solution uh, and in a sieve like gel, and then we apply the electrical current. So those molecules that could be separated based upon their molecular mass, that molecular weight, and then we can uh, quantify them. We can characterize, we can see the conformations of those, and then we can study their nature and molecular, the molecular weight as well. So we use the electrophoresis for separation of those charged molecules based upon their mass. Then we will see that what kind of electrophoresis, for example, we have, for example, horizontal gel electrophoresis, agarose gel electrophoresis, which we have been using for DNA, which is a routine lab technique for quantification of DNA, as well as studying, for example, restriction digestions, as well as quantifying DNA, genomic DNA as well. Analysis of DNA, either it is, for example, uh, intact or it has been degraded. Then the vertical gel electrophoresis. In vertical gel electrophoresis, we have SDS page electrophoresis as well as native gel electrophoresis. So SDS page electrophoresis, that is used for denaturing gel electrophoresis, while native gel electrophoresis, that is used for studying native conformation of the proteins. And that also can be used that ultimately, if we want to purify the protein, we can use it for isolation of the native form of protein. Then troubleshooting, we will see also that how we can, for example, stain the uh, SDS page, the vertical gel electrophoresis, that two methods most widely used for the, especially the Comazine brilliant blue that we have been using for staining, while we have been, we could use also like silver staining, however, that is a bit expensive as well as bit longer as well. So all these, then we will see the applications of electrophoresis, that what are, for example, different applications of electrophoresis. So let's start from the electrophoresis, that is the transport of the particles through a solvent by an electric field. For example, when we use the molecules, charged molecules, uh, and if we apply the electric field, the macromolecules, they will move under the electric field, and then we can use that technique to distinguish the molecules, to identify the molecules based upon their charge and mass ratio, charge and shape, and also we can use it for determination for molecular weight of proteins or DNA, and then quantify as well. Theory and basic concepts that a separation of large molecules like DNA or protein that depends upon two forces, charge and mass. So when biological samples like DNA or protein are mixed with buffer solution and then applied on gel, the two forces acts together. That the electrical field, when you electrical apply the electrical current, one electrode that would repel while the other that would attract them. Uh, Repel the molecules while the other electrodes simultaneously attract the molecules. So the gel material acts as molecular sieve, and then uh, the molecules are forced to move through these pores of the sieve, and the rate of migration of those molecules that would depends upon the strength of the field. That if you increase the strength of the field, that if you increase the voltage, that the separation that will be faster. However, that would result in production of more heat, and that would result in uh, diffusion of bands in gel. That production of more heat that may also accelerate the degradation of the proteins as well. For example, if there are enzymes, so that may result in degradation. Then size and shape of the molecule, that size that lower the molecular weight, more rapidly it will move, while higher the molecular weight of the molecule, the slower it will move. While ionic strength and temperature of the buffer, which is in which the molecules are moving as well. So these are the three factors that will influence the migration of molecule in the sea. Then the frictional resistance of the sieve that will slow down the movement of the charged molecule of, like DNA and protein, and the frictional force that will that is a measure of the, for example, hydrodynamic size of the molecule, that shape of the molecule, the pore size of the medium, and viscosity of the buffer. So hydrodynamic size of the molecule, that, for example, superchiral DNA that will move faster, while the linear DNA that will move slower. While the shape, that is same, that the shape of the molecule. Then pore size of the medium, that if the pore size would be bigger, that the molecule that will move faster. But if the pore size that will be smaller, then the molecule that will move slower. Then mobility depends upon the frictional coefficient, which in turn depends upon the physical parameters of the molecule. Therefore, mobility gives the information about charge, size, and shape of the molecule. So that would give the information about size, shape, and charge of the molecule. The current in the solution, as we just discussed, that if you, for example, increase the, for example, the voltage, that would result in accelerating the electrophoretic separation, but increasing in voltage, that would also result in the production of more heat as well. So if that will result in heat, although the migration that would be accelerated, but that would also result in production of more heat, and heat that may denature the molecule, that may result in diffusion of the molecule, and the thermal stability of the molecule, that, for example, if there are enzymes that may degrade it. Then decrease in buffer viscosity, the production of more heat, that would result in decrease in buffer viscosity, and reduction in the resistance of the media. So the separation, if you increase the voltage, that the acceleration of the molecule, that movement of the molecule, that would be faster, but separation that will be resolution that will be low. However, if you decrease the voltage, the movement that will be slower, but the resolution will be good. Then types of the electrophoresis, one of those we just discussed, the agarose gel electrophoresis, that is horizontal gel electrophoresis, which we use for the DNA, for quantification of DNA, for example, restriction endonucleases, when we digest the DNA, analyze the uh, cloning of insert in vector, plasmid vectors. So when we digest those plasmid vectors, we can use the agarose gel electrophoresis for analysis. Then polyacrylamide gel electrophoresis, in which agarose is a seaweed, and that is not useful for proteins. So polyacrylamide gel for proteins, usually the polyacrylamide gel electrophoresis that is used, and that are made by the polyacrylamide gel that is made by acrylamide and bisacrylamide, that polymerization of the acrylamide and bisacrylamide. And in this one, that APS and TMAT that are used for the polymerization um, agents. Then those particular polyacrylamide gel, then this particular gel, polyacrylamide gel electrophoresis, that is further divided into non-native phase and STS phase. So STS phase is denaturing, while the native phase, that is non-denaturing. So polyacrylamide gel electrophoresis that could be used for both DNA as well as proteins. So polyacrylamide gel acrylamide components are acrylamide, bisacrylamide, tricyclic, TMAD, and APS. TMAD here is the acrylamide, and this one is bisacrylamide. So after polymerization, the structure would look like this one. That polymerization would take place, and then that would result in formation of gel, that would result in formation of sieve, and the the thickness of the acrylamide gel, that is it's just a few millimeter thick, while the agarose gel, that is quite thick compared with uh, polyacrylamide gel. Pore size of the polyacrylamide gel, that depends upon the concentration of the acrylamide. That if you increase the concentration, the pore size would be decreasing, while if you decrease the acrylamide concentration, the pore size, that will be increasing. So depending upon your particular purpose, that the size of the your the protein, you can use particular concentration of the percentage of the gel. We will see in the next slides that there could be different percentage of gel, which would be again the components, the volume of the components, that would also be given. That, for example, this particular percentage of gel, you can use this much volume of all those components, and you can prepare. So APS and TMAT, those are the polymerizing agents of the acrylamide and bisacrylamide. Uh, so TMAT and APS that are required for polymerization of the uh, acrylamide and bisacrylamide. So here you can see 7%, 10% acrylamide concentration, as well as 12% acrylamide gel concentration. So SDS, SDS actually is a detergent, and that would result in denaturation of the uh, protein. So we will also use beta mercaptoethanol, uh, that would uh, reduce the disulfide bonds that are required for um, the tertiary structure of the protein. So tertiary structure of the protein, you know that hydrophobic interactions, disulfide bonds, ionic interactions. All these are stabilizing tertiary structure of the protein. So when we use, for example, beta mercaptoethanol, that reduces the uh, that reduce the disulfide bridge between, especially like cysteine and uh, cysteine residues. So what happens when we treat with SDS? So that would result in denaturation, and then, for example, the treated produce proteins assume the rod-like structure that all carries the same net negative charge. So when you will electrophrase them, the gel, the protein will migrate towards the positive electrode, then separated depending upon uh, their molecular weight. So that is that we also heat. So heat results in denaturation. SDS that results in denaturation, and beta mercaptoethanol that is used to remove the disulfide linkage between the proteins. So native gel, as we discussed that, SDS page that is denaturing gels, while native pages are used for studying conformations of the proteins, because that does not uh, denature the protein. So that could be used to study the conformation, self-association, or aggregation and binding of the other proteins or compounds. While it could be also used to recover the proteins in their native forms. Then there are another continuous and discontinuous. For example, continuous, that means that there could be only resolving gel that would be present, and you can use that for separation of the protein. While discontinuous, that there are two kinds of gels. One is stacking gel, and the other one is resolving gel. So stacking gel that has a larger pore size, and that means to stack the proteins, and then the protein would go from the stacking gel to the resorbing gel, which will separate the protein depending upon their level of weight. So here you can see, for example, the stacking gel above, while on the lower side you can see the resorbent gel. So the stacking gel is on the upper side. So first we make the resolving gel, and then we make the stacking gel on the upper side. So here are the, for example, uh, the pH of the stacking tricyclic pH in the stacking gel, that is 6.8, while pH in the resolving gel tricyclic pH, that is 8.8. While in glycine, that is running buffer, that's pH is 8.3, while tricyclic pH 6.8, and that is sample dilution buffer. So in stacking gel, the pH is 6.8. That negatively charged form is disfavored, and average electrophoretic mobility of the protein, that is low. However, in separating gel, the pH is 8.8. Negatively charged is favored, and the electrophoretic mobility in resolving gel, that is higher. So, for example, here you can see that as glycine from the electrode buffer enters the stacking gel, the boundary between the glycine and chloride forms. The chloride will move faster, while the glycine would follow that one, while the protein that would be present between the center of these two. So here you can see that the protein have mobility between glycine and the chloride. So while when they enter in the resolving gel, so the chloride that will have higher mobility, that would followed by the glycine, and then that would followed by the protein. So here are the apparatus of electrophoresis. So you can see the vertical gel electrophoresis, and then you can see the plates of the vertical gel electrophoresis and comb as well. So these are the that electrodes plates and the electrophoresis tank. Then these plates, they have been shown by for preparing the gel, that how you can prepare the gel. And then here, for example, you can see the gel that is visible, and then placing the gel in the electrophoresis tank. And you can see here, for example, gel have been placed in the electrophoresis tank, and then there will be two chambers. One is outside, while the other one will be the inside. So you will fill both these chambers, and then you will load your samples. Then visualizing, for visualizing, as we discussed, there are two methods. One is combustible, while the other one is silver staining. Silver staining, that is more sensitive, but that could be hazardous, as well as that is time consuming. But commercy blue, that is simple, commercy brilliant blue, that covalently binds the proteins, and that results in visible blue bands of proteins in the clear background of the gel. So both of these you can use. However, generally we use commercy blue, brilliant blue. So here is the procedure of the commercy blue, that how we prepare the commercy blue, and then how we stain, and then we use de-staining solution for the commercy blue. And then silver nitrate procedure for the silver nitrate, that is fixing, then incubation, then washing, then silvering, and then again washing and developing, and then stopping, and then finally drying. So this one is the different reagents that have been used for these silver staining. Well, these are the volume and time. So the silver stain gel, that is silver staining procedure, that has higher sensitivity, and the gel will look like this one. That is silver stain gel, while the commercial blue stain gel, that will look like this one, that the protein will be visible as blue bands on the clear background of the gel. So here you can see the ladder of the protein, while these are the different proteins. Applications that of electrophoresis that we can use, we can determine the molecular mass and effectiveness of the procedure to purify specific organelle protein or polypeptides, uh, while we can also use to identify, for example, quantify the proteins as well as DNA using electrophoresis. Thank you so much. If you